Alright, let's do a mic check testing. Alright, we're good on mics. So hello everyone, my name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and welcome back to Cytometry in R. So this recording is covering one of the extended bonus walkthroughs tied with week 12 spectral signatures. Basically, this one's not meant to supplant the week 12 material, but it's meant to complement, going in additional depth to some of the aspects that are being covered, but then also more importantly, showing you how the different steps were done in the background, more aimed at if you're trying to implement it on your own without having to go as far into the detailed weeds about what's actually happening in terms of each individual line of code. So it's a little bit different from what we are normally experiencing. We're giving this format a little bit of a try. We'll see how it goes as we move forward. So for this particular walkthrough, I'm going to show you how to download datasets from ImmPort, namely the dataset that was used during the week 12 through the week 14 part of the Cytometry in R course, working with the full entire FCS files rather than the small downsampled FCS files that were actually included for the course. So, this section of the course is primarily focused around raw spectral flow cytometry files. So during week 12, we are learning how to extract out and characterize fluorescent signatures that are present within our unmixing controls, both single color and unstained. This is being followed up on week 13 by learning how to compare these signatures with each other, which allows us to predict how they will interact in the context of a full spectral flow cytometry panel. And then finally, in week 14, we take these fluorescent signatures that we have derived from our mixing controls and complete the circle using them to unmix full stain samples. So, due to Cytometry in R's course use of GitHub, we are not able to include the full-sized FCS files due to the individual file size sharing constraints. The datasets that are used through week 12 to 14 consequently are downsampled to get around this issue. However, working with complete datasets can provide additional insight, as well as more closely replicate the experience of working with your own datasets. So this walkthrough is basically the steps that would be needed to both retrieve and process the original files, as if you were trying to use them also from the ImmPort repository. So ImmPort, or otherwise known as Immunology Database and Analysis Portal, is an open access platform for research and data sharing, primarily intended as a repository for datasets that were generated as part of either the NIH or NIAID funded grants here in the United States. It includes many different types of immunological data, including both conventional, spectral, and mass cytometry datasets. So we are slated to cover how to retrieve the data programmatically from ImmPort via the API later on in the course. However, in the meanwhile, this walkthrough will cover how to access and download the individual datasets via the website portal. This can be a bit of a challenge for those who are uninitiated, so we'll depict one of our typical workflows that can then be used later on. So, to start off, we first need to navigate to the ImmPort repository. So the landing page at the time of this writing this walkthrough can be seen in the screenshot below. And the main thing we're going to need to do is go up there to that little login tab in the nav bar. So if this is your first time using the ImmPort repository, you'll need to create a free account. So the simplest way, just go up there and click on the login button that's located on the upper right hand of the ImmPort website's homepage. Once you go ahead and click on login, what you're going to see is you're going to enter a login page, but it also includes the option to register for an ImmPort account. So go ahead and select that option to go ahead and register for that account. This will then proceed to a page where you'll be notified about the next steps and also the need to accept ImmPort's term of use, which is linked above, before you're able to actually access the datasets. So in this case, you would then select Continue. At that point, you're going to be prompted to fill in additional fields, both a username, name, email, password, all that basic registration kind of style information, password retrieval questions, etc., etc. So I didn't provide an example of what it would look like to be able to fill this out, but you would just fill this out with your own equivalent information. Next, you're going to proceed through several additional steps before you ultimately end up completing your account registration. At that point, you're going to get returned to a login page. Now, if you're registering for the first time, please just go ahead and take some documentation screenshots of those last couple pages. Obviously, leave them blank or white out any important personal information, and go ahead and post them to our issue page. So I had already registered for my ImmPort account long ago, so I wasn't actually able to get those last few screenshots. So if anybody wants to contribute them back, I will update the website page accordingly. Once you have completed the registration process, you'd then be able to log in again for the first time. And when you log in for the first time, or you log in for the 20th, 30th, 100th time, they're all very similar as far as the process goes. So once you log in, you're going to go through a two-factor authentication process where ImmPort will email you a code. You need to access your email, provide the code to be able to get past the next screen. So this is an example of what that ImmPort email is going to look like, providing you a one-time login that would expire within 10 minutes. You just basically copy it, head over back to the login page, provide the input code, and then select Submit at that point. And with that, congratulations. If you haven't yet done so, you're now registered and also logged into ImmPort, and you're finally able to access and download the datasets for this particular example. So at that point, once you're logged back into ImmPort, you're still seeing very much similar in terms of the home page as far as the layout goes. As I've mentioned, ImmPort can contain many different forms of immunological data, including conventional, spectral, and mass cytometry datasets. So, when you're actually trying to figure out what we're going to use for today, the data that's being used for the week 12 to week 14 of cytometry in our course's examples is originally from the SDY 3080 dataset. This is the dataset that I generated as part of my 2025 paper that was in Frontiers in Immunology. It basically looked at cord blood innate-like T-cell responses in neonates that were born to healthy women and women that were living with HIV. As of the time of this course, it is one of the few spectral flow cytometry datasets currently on ImmPort that is not just the already unmixed files, but it also contains the raw data that you would need to do actual unmixing of the files over again. So, to actually go out and figure out where it is, once you've logged in, basically you can look up the accession number, which is the SDY 3080, within the search bar to be able to get to the study. Similarly, if you're interested in a particular search term, like HIV, like neonatal, any of those combinations that might be in the title, you could use that and look in the search bar to see and navigate to those kinds of data sets if you're interested in them. Once you end up getting to the search results page, if you look up an accession number, generally this is the only one that you're going to be able to see. And this is the situation like you found here. Alternatively, if you search for a more generic term like flow cytometry, it might come up with a bunch of other variants across the board. So to actually go ahead and get started, you just need to go ahead and click on the study accession number. It's located on the left side of the little drop-down to actually proceed to that study-specific page. So you would just go ahead and click right under here under study, and it would proceed to go and show you the page itself. So now the page itself will show you a little bit about the details about what the study is, the sign, demographics, all that other information that makes ImmPort valuable, because it's the combination of everything that is what people are trying to leverage for more big data, big science kind of questions. But in the context of what we're trying to do for the course, we really just need the FCS files. So the way we can go about doing that via the website instead of programmatically is under the downloads options that you see in the middle right of the page. You would just select the study package for the web option to access that information. This would then take you to the studies data folder. So ImmPort studies in general contain additional layers of metadata, which assist in the ability to reuse the data for later purpose, long past their original intent. So these are going to get covered in greater detail later on in the course, but for now we only want the results folder. So as we get to this little internal page, we're going to go search for those results files, if you're trying to find like additional information about the study, that's oftentimes going to be under protocols as far as the individual folder goes. And once you're at that results folder, ultimately it depends on what kind of data sets are included for the particular study. In this one, there was only a flow cytometry results subfolder, so that's pretty much your only option to click on. If there had been ELISA data or other forms of data, they would have their own specific folders, so select on the equivalent one that you actually want. And at this point, you've reached a page where all the individual FCS files are listed. So to actually proceed, you can click on the different tabs a couple times just to make sure you're reorganizing the files in terms of largest to smallest, vice versa, just so you have a general idea of how large the data sets are that you're trying to download. And then once that's done, I always just recommend going ahead and selecting the Show Me As Many Files As I Possibly Can option, which in this case is going to be 200. So at this point, you can see when I'm visualizing going from 50 to 200, there's over 973 items present within this folder. You can see on this side, when I'm looking at some of these speed controls, their original file size is about 1.43 megabytes, so low large, but still compatible with GitHub. But then as I start looking at the others, they get substantially larger. So for the week 12 to week 14 examples, ultimately we wanted to do the unmixing controls, so I ended up grabbing unmixing controls from the first 2 experiments within the SDY 3080's dataset, which I think was up to 15 experiments in total. So that very pre-experiment, the ILT_00, consisted of a set of bead single-color unmixing controls that were acquired before the experimental series start. This was mainly as a hedge, just in case any of the individual cell controls didn't have enough events to make a proper signature. And then the second data set grabbed for cytometry in R was the actual first experiment where everything was a cell single color control. And additionally, there's often a lot of additional unstained samples, namely things that are specimen-specific, condition-specific, unstained unmixing controls. And then I also went ahead and grabbed a few of those full stain samples just because they will be encountered during the week 14 of the course. So, if you're trying to select multiple FCS files on ImmPort, you can do this by clicking on the checkboxes that are located on the left-hand side of the screen. So as you can see here, I'm selecting a few of the bead controls, a couple of the cell controls, and then at that point you can see as I add things, they get added up above to the View Cart tab. So, once you have selected the specimens that are of interest to you, you can proceed to click on the Download Cart option just to see how you can actually export them. So once you select on Download Cart, what you're going to get is a pop-up, and it's basically going to ask you, do you actually want to proceed and download these ones? So in this case, I would try to go ahead and grab everybody from the very get-go. So I grabbed all the single color controls for beads, all the single color controls for cells, all the respective specimen condition specific unstains for the first experiment, as well as some of the full stains. So ultimately I had about 15 gigabytes of data that I was attempting to download. So you can go ahead and select next, at which point a preparing download pop-up screen will typically appear as your folder gets zipped and prepared for export. So this is typically what you'll see as it starts to do this. And as you are waiting for this to do, ultimately, if it's retrieved successfully, you're going to get a little prompt that pops up to save the folder to a desired location on your computer. At that point, you would just proceed and extract out the contents for subsequent use, just like you would do for any random zip folder. So in this case, for this particular data set, I'm saving it under my SDY 3080 folder, and then I'm placing it within the data folder subfolder that's present, just to keep things kind of organized. So at this point, basically, you would see I'm going to try to extract out the contents of my download, and then correspondingly place them in the correct locations. So, occasionally, especially when you're trying to download via the web, ImmPort will end up failing during its attempt, and it will give you an email that's something like this one popping into your inbox, basically saying your attempt at download of the zip folder failed, and will generally give you something about zip file size would require zip64 extensions. So, and then it gives you access to see the log of the failure notice. So, from my limited testing this week, it often appears that this is when the combined file download size exceeds a certain size. So when the total files that are being downloaded are under 1 gigabyte, generally this is going to work, but when it's past that point, it needs that extension to be able to generate the zip folder and the download to proceed normally. At which point, you would get a notification that looks something like this, where everything is cooperative with you and you're able to proceed and download. Alternatively, though, if you don't have that zip extension and you're struggling and you just need a couple of the files, you can download these individually by just clicking on the individual download tab right next to it and then proceeding to download accordingly. So if you want to just download individual files without having to select multiple, it's going to be this little download arrow icon that we see right here. So, beyond the manual download options, there are several other ways to more efficiently download the files via the API, which is outside of the course of this small walkthrough. We intend to cover these in greater topics in the future just because they are particularly valuable if you're already working in R and you want a more simplified manner, or if you just want to be able to investigate the metadata or identify specific files that you want to copy over ahead of time. So, for this walkthrough, we are going to go ahead and organize the retrieved files in the following manner. This is going to be held consistently for both the ones we end up downsampling down to actual size, but then also for all the subsequent lessons in this particular section. So we want to basically simplify our ability to quickly switch between FCS file types, namely single color beads or cells, unstained, full stain, so when working on the various downstream applications that we plan for this section. So this is how I'm ending up organizing. So underneath my actual working directory, which would have been the SDY3080 for this example, I have my data folder, similar to what we've been doing in terms of course organization. There's another SDY3080 folder at that point, but you can see I'm creating some subfolders for full stain, for single color cells, for unstained cells, and then for single color beads accordingly. And then I just make sure when I'm extracting and unzipping these folders, the respective files get placed accordingly in their correct locations. So a couple things to note, we also should just go ahead and double check what the file size of these individual cell single color controls are. So once they've been extracted, you can change them in your window display depending on what your operating system is. And as you can see, some of these single color cell unmixing controls are rather large. So the largest here for the PD1 was 112 megabytes. And also, if you looked at the single color bead controls, which only acquired up to 5,000 events, you can see it's much fewer across the board with only 1.4 megabytes in the total file size. So this dataset also contains specimen match, condition match, and stain samples that were acquired to allow autofluorescent signature profiling. The amounts of cells would vary depending on how many cells were left after the cord blood was thawed, and after the full stain samples were aliquoted accordingly. But as you can see, some of those files became pretty large. So the largest unstained file that's present for infant 182 was about 322 megabytes across the board. So, likewise, as anticipated, the full stain samples are going to be large, given that over 3 million events were acquired in order to have enough events to profile cord blood innate-like T cells. So as you can see, the largest of these files was over 800 megabytes just for that individual raw FCS file. So these are some of the caveats that you would encounter if you're working with your own datasets, but then also if you're trying to download somebody else's dataset that has been placed on a common repository, either ImmPort, Zenoda if you're more on the European Union side, or if the repository comes back to life in the next year, some of those similar struggles as the different people involved with the ISAC and Cyto in general are trying to keep the repository up and running. But if every time you upload a new data set, you have to deal with this kind of amount of data, it's a little bit of a challenge across the board. So, all in all, this is a relatively quick walkthrough. We briefly covered how to register for an ImmPort account, how to access individual datasets, and how to download the FCS files that were used for the Cytometry in R course during the weeks 12 to 14 session. So beyond what we showcased here, there are additional repositories on ImmPort that contain rather interesting cytometry datasets. Showcasing how to identify, access, and download these in a more efficient manner will be the focus of later on in the course. You are, however, more than welcome to investigate on your own until then and see if you find anything interesting that you would rather actually play around with. And with that, that's all I have for right now. I will record the next walkthrough for this evening in about 30 minutes. Covering the actual subsequent steps when it comes to cleanup gates being implemented for both the cell single colors, the bead single colors, and the unstained cells, getting them all set up into those intermediate GS folders that could then be used for the actual drawing of the signature gates based off of the peak detector, kind of similar to what we saw during the week 12 of material. So I'll be back shortly with that one, and if not, have a good evening, a good day, wherever you are in the world, and I'll see you next time.